Thank you. Oh, it's wonderful to see all you here to listen to fish, about fish stories. That's really exciting. I'm really pleased to be able to introduce Melina Hale, who's the William Rainey Harper Professor, and as you know from reading, the Vice Provost for Academic Initiatives. I'm really thrilled to be able to introduce her tonight. She's not a stranger to the MBL, as most of you know, and her time here started back in 2000 when she was a postdoctoral fellow working with Joe Feccio at SUNY Stony Brook. And if I could have the first slide, because we do need evidence. You know, everybody claims they've been a grass fellow, but really the evidence is whether they've worn a grass skirt in the 4th of July parade. And I'm going to leave the microphone, but you can see this is Melina right here. This is 2000. This was a time when the Grass Laboratory was on the 3rd floor of Lilly Building, this building, in the lab of Harry Grunfest, the lab that Harry Grunfest used. And her year and a few years after were in that lab before it moved over to the what's called the Rowe Building now. And again, the evidence here, although it's a little fuzzy picture, is this is Melina here, Eve Marder's up there. But the grass skirts are evidence that she actually was here. So she knows the MBL well. She worked on the lumpfish here when she was a grass fellow, and I had the pleasure of interacting with her then. She, since then, has done research here at the NXR, the National Xenopus Resource, run or directed by Marco Horb, and she's on the external advisory committee for that unit. More recently, as David mentioned, she's interim co-director along with Neil Shubin at the MBL. And her leadership skills are remarkable. One of those skills is her ability to communicate, and you'll witness that tonight. But she's used that ability to communicate in a clear and transparent fashion to help the MBL, the MBL community, all the way from students to faculty to staff to the scientists to the administration to talk in a clear and transparent way about issues that existed, and she has kind of shepherded us through what I consider a kind of a shaky period, and she's done a remarkable job. And I think on behalf of everybody, like David mentioned, I think we should thank her really sincerely, so I'm going to applaud again. A second leadership skill of many is on the next slide. And so the directors have always led the 4th of July parade, and she did it with aplomb. So she's shown a positivity that I think has been very important to the MBL over the past year. Her research is hard to describe, but I think 2 words kind of encapsulate it, and integrative and comparative. And so it's appropriate that she was elected, she's president-elect of the Society of Integrative and Comparative Zoology. She is very much interested in taking a look at a number of diverse organisms and integrating the information on how they move and how the neural circuits underlie that movement. And she's going to share this with us tonight. So with that, I give you Melina Hale.
Well, little did I know that wearing that grass skirt so many years ago would prepare me for another 4th of July many years later. The MBL has been formative in my life as a scientist, and so it was an absolute privilege to be able to support it over the last year. And everyone was just so welcoming. You know, when I came, although I benefited from all the people at the MBL, MBL is a place. It's an organization. And what we heard when we started, Neil and I, was that, MBL is a place, and you have to worry about the buildings and all that kind of stuff. But it's really a community, and that's what's special about it. And anyone I talked to, that became evident how special it was, both in that communication of how everyone cares for each other, and also everyone's willingness to do anything they could to support this institution. So I would just like to say it's been just such a pleasure to work with all of you and our colleagues who aren't here tonight. It just really is a magical community. So we'll turn to the subject of tonight's lecture, which is fish. So maybe this will give you something to think about at all those wonderful fish dinners, or when you're out on boats catching fish this summer. My lab is interested in how animals move, how they move mechanically in their environment, and how the brain and the spinal cord control their movement systems as well. We study fish as our model organisms for a number of reasons. I want to work on vertebrates. I've always worked in vertebrates, and fish are the most diverse group. There are about 30,000 species. Within those 30,000 species, there's exceptional diversity. So we can not only use big families to test evolutionary questions, there are lots of really interesting specializations of fishes that we can use to get to particular questions of interest in this broader question of how animals move. So tonight I'm going to talk about two of our main interests in lab, and let's see if this works. Both of these projects involve these fins right here that are called the pectoral fins of the fish. They're paired forelimbs. They're homologous to our arms, forelimbs of dogs, turtles, other tetrapods. And they're important in a lot of fish for swimming and actually for other behaviors as well. This is a bluegill sunfish from Illinois, and it's been one of the models we use in our work. The biggest system we use in my lab, though, is the larval zebrafish. So zebrafish are a genetic model organism, which is terrifically helpful. But for my lab, they really demonstrate an even more important specialization that's critical to our work. And that's that they're transparent when they're larvae. So at about 3 millimeters, which is about 5 days of age in the larval zebrafish, and you can see the whole animal. We're looking at just a section of it here that corresponds to about that area of the animal. You can look right into the brain and spinal cord in the whole live, intact animal. In fact, I had to put this little schematic of the pectoral fin on the image because it's completely transparent and you can't see it. So I was trying to point it out when I was practicing and just gave up. But the pectoral fins of the zebrafish are here, and we'll talk about this tonight, in particular as a model for understanding fundamentals of how limbs are controlled and move. The transparency you can see here. If we take away the muscles and look at them in fluorescent light, we can see the nerves beautifully. We can see the brain and the spinal cord. I won't talk a lot about the neural circuits in the CNS, the central nervous system, tonight, but that's one of the big focuses in my lab. Another reason, very different reason, that we're interested in fish swimming and in fins in particular is to inform the design of autonomous underwater vehicles, so engineered devices. And so this is one fish. Actually, if you look at it too long, you might start feeling dizzy. You can see the sway. This is a robot that a colleague of mine, James Tangora, and another collaborator, George Lauder, and I have been working on. This is a poorly designed robot. As you saw, we were shifting from side to side. What we're doing in our lab is using examination of fish movement and of their neural control in order to better inform how these devices move in their environment and also how they're controlled. And I'm going to come back to that towards the end of the talk. So what do fish do with their fins? My title said swimming and walking and flying, and they honestly do all of those things. So just to give you some examples of how wonderfully diverse the movements of these animals are, these are juvenile reef fish. The group is Pomacentrids, but when they grow up, they're pretty little fish that you might find in an aquarium. Right now, at least when I took the video, they were swimming in a flow tank, so basically a treadmill for fish. And you can see that at slower speeds on the top, they're alternating their fins, left, right, sort of like our walking gaits. When they go faster, they actuate their fins together. They get more power that way. That's more of a bound that you might see a dog performing when it's running very quickly. So fish show not only the same patterns of movement, but the same types of gait transitions that we see in mammals or in other tetrapods. On this side of the screen, you see lungfish, and lungfish are actually more closely related to us than they are to the fish on the left. And they actually do physically walk on the bottom. So you can see they're not quadrupedal, they're bipedal, they just use their hind limbs. And in this bottom image here, you can see that they actually lift their body off of the bottom of the bottom substrate. This is actually a really, Neil and I who've been working together this year, have collaborated for quite a while, and this was a fun project to do with him. We could actually inform, by studying fish and the control of their gaits, some of the hypotheses in the literature of when organisms may have come onto land. So I'm not going to talk about that today, unfortunately, but fascinating fish that walk. So let's start with the zebrafish, which is, as I mentioned, the main animal I use in the lab. And I want to highlight Dean Thorson and Matt Green, who are former graduate students of mine, who did a lot of the work that will be coming up next. This is a zebrafish in frontal view, so we're looking at its head. And again, remember, these are 3 millimeters long, so we've got about 1 millimeter across here. Tiny animals that we're looking at on pretty high-powered microscopes. So what we were interested in doing is really using these limbs of the zebrafish to see if we could figure out, how the limbs are working, what they're doing, and how they're controlled. Not only are zebrafish transparent and have genetic tools that I mentioned before, but we could also do some wonderful fluid dynamics approaches where we could use computational modeling to play with these animals. So we could go all the way from the behavior of the animal and its physical environment through looking at its muscles and its nerves and its neural control physiology to really getting an idea of how this animal works. The end of the story will be it works nothing like what we thought when we started. In fact, it's doing something very different than we thought. But that's the fun of discovery and what we do at MBL. So it's a really great project. So here's a more intuitive view of the zebrafish on the left. And you can see start this movie, the coordination of the limbs and the axis here. And note that compared to this salamander, which a video courtesy of Miriam Ashley Ross, who's also a grass fellow in a different year, the zebrafish pattern of movement looks a lot like a walking salamander, not much like a fast swimming salamander where the limbs aren't working for the animal to generate propulsion. So this is the behavior that we are interested in understanding. The big question that we started with when we first started showing people images and videos, we got, well, are you sure they're actually even doing something on their own? Maybe they're just passively flapping because they're larvae. They're not formed yet. They're working to get to the adult form. Maybe these are just immature, tissues that are off the body and they're moving passively in the water, but they aren't doing much. Or even if they're active and moving, do they really have a function for the animal at this stage of development? So we started asking those types of questions. Oops. So the first thing we had to do was go in and actually see, is there muscle in the fin? And is it innervated? And is it actually being activated during movement? So here's that the image from a few slides ago. And just to place this green dot is within the fin where the muscle is, and it corresponds to the muscle fibers that you're seeing here. Right? So each of these bands is a single muscle fiber. And they're located at the base of the fin, right here. It's one muscle, and it's one muscle, sorry, it's two muscles. It's one muscle that brings the fin up, and one muscle that pulls the fin back to the body. Abduction is extension, moving the limb away. Adduction is moving it to the body. So those are called the abductor and adductor muscles. As you can see, in addition to there just being developing muscle there, we also have extensive motor neuron innervation. So all these white lines coming across are motor neurons that are extending to innervate this muscle. So just by our first anatomical studies, we could see the fin was set up. It had the anatomy to actually produce movement and to potentially function in locomotion. Just because it has the anatomy doesn't mean it's working. So the next question we had to ask was, physiologically, are these nerves actually firing to stimulate the muscles in an appropriate pattern for movement? And just step back for 1 second and look at this movie, which is a lot like the salamander, in that when the fish swims fast at the beginning of the video, it'll loop back to that in a minute, the fins are tucked. And when it slows down about here, you start to see fins extend. You know, this suggests clearly that the fins are probably actively controlled to begin with, but it also is very reflective of the salamander movement. At fast speeds, limbs are tucked. At slower speeds, it was walking. So my student, Matt Green, was actually the first one to show these gaits and how they worked. And I won't go through the electrophysiology, but he did a lot of patching to actually show that during fast swimming, we saw this rhythmic activity of the abductors and the adductors, shown here. And during that faster swimming, which had been hypothesized to have just passive movements of the fins, in fact, we found active tucking of the fins against the body in fast swimming, and inhibition of those muscles that would, or the nerves that would control the muscles that pull the fin away. So clearly this is sophisticated movement, and the gaits are similar physiologically, behaviorally to other organisms as well. I'm not going to talk a lot about the detailed circuits in the spinal cord that we're actually working on quite a bit in my lab now, but did want to tell you another part of Matt's work that, again, using the benefits of the larval zebrafish, allowed us to figure out where these gaits are controlled in the brain. And so with the larval fish, because they're so small, and actually they respire across their skin, they don't have functional gills yet, so they just uptake oxygen over the skin and release gases. You can actually do a lot of crazy things to them, like cut off their head and watch the spinal cord and body swimming. And then, sorry, it's a little graphic, but for people like me who work on spinal cord, it's an amazing benefit of the organism. So what Matt did was actually go into the brain to do a first step of, well, can we figure out where the descending control is that's causing these gaits to occur? Is it coming from the brain? Is it spinal cord? If it's in the brain, where is it? And so he was able to do various transections, having a whole brain, basically cutting these tracks between the midbrain, which is supposed to house the locomotor regions, and the hindbrain, and then cutting at different locations in the hindbrain. And what he found was that with even if you took off the midbrain, the fish could do all of these gaits. The descending control, particularly that seemed to be important for that faster gait where the fins were actively tucked, was not due to input from higher levels of the brain. It was due to rostral hindbrain. When you cut back further, it really took quite a bit of transection cutting back until you could change that gait pattern. And what would happen is we lost the fast gait, but we retained that slow rhythmic movement and the coordination of the fins and the axis. So what he came away from this saying is both the locomotor, the descending control for fast gait is not coming from midbrain, it's coming from farther back in the brain, which is unexpected. But also these really, what look like very complex rhythmic coordination does not need even rostral hindbrain to conduct. So as I said, I'm going to diverge from this right now and not talk about circuits, but I'd be happy to at a later tonight or at another time. I wanted to go back to my bigger story, which was we have these fins, we don't know what they do. We don't know if they are even functioning in these animals. We figured out that the anatomy looked good for it to be a functional system. Physiologically, they're working. Behaviorally, it looks like they're moving to generate locomotive forces. But the ultimate test was, actually, do they matter in locomotor performance? And so to get at that hypothesis, we used the genetic tools in zebrafish, basically injecting a morpholino to block translation and early development to take the fins off the fish. So on the left, you see a normal zebrafish with its fins intact, and on the right, a fish that's basically normal, same size, same stage, developmental processes seem normal. It just doesn't have pectoral fins. And what we found was there is absolutely no difference in their behavior. So here we've been developing this locomotor model, thinking how important they are. Performance, it doesn't matter. It didn't matter for thrust generation. It didn't matter for pitch or yaw or roll of the animal. That we could not find any biomechanical reason that the fins were functioning in locomotion. So we started going back to the older literature and looking at details and papers and trying to think out, well, what is going on with these fins? We saw a very short notice in papers by Batty and Blackster, two wonderful ichthyologists who worked in larval fish, who suggested that maybe the fins are respiratory organs and not locomotor organs in larvae. As I mentioned before, there are no gills at this stage. It's all cutaneous respiration. So we decided, okay, well, let's hypothesis. I started with a simple fluid dynamics experiment, putting dye in front of the fish and seeing what the fins did with the dye. When fins are propulsive, they tend to generate nice vortices, showing you can calculate force production. These fish didn't do that. For these small animals, when you think about it, because of their size, when you look at the fluid dynamics of the water, to them it feels more like honey. It feels thicker, more viscous. And so they didn't generate vortices. Instead, they folded the fluid along the side of the body. Folding like this is a wonderful model for mixing fluid along the side of the body. Where do we take up gas exchange? Where is gas exchange? Where's oxygen uptake occurring? Right along the side of the body in that trunk region. So this was exciting. We went on to do a lot of fluid dynamics with some colleagues at Northwestern and found that when we just modeled the behavior in their flow models, we saw that same pattern. So the fluid is just marked in these colors, so you can see it passing along the fin. And if you go from the beginning of the trials to the end, we see that nice fluid mixing along the body. The fish are bringing water from far away from the body in, presumably oxygenated toward the body, and moving deoxygenated water away. As we investigated this further, it became obvious that of course they have to do this. So we just happened to capture this wonderful trial of a fish moving through a dye stream. And the notable things for me as a fish biomechanist, as you see it pulling through, right, you see this area that's clear right along the sides of the body. What that's showing you is the boundary layer of the animal, and it has two effects to it. All this water is entrained. It's coming from over here and clinging to the fish and moving through the dye with the fish. That entrainment of water is adding a lot of added mass, so it's making it harder to move, but it's also stagnant against the side of the body. It's water that's moving with it and where you won't get good circulation. So Matt Green did some really nice experiments in the end where we changed the dissolved oxygen levels in the water as a final test of whether the fins seemed to be involved in respiration. And what he found was that, to make a long story short, was that when there was low dissolved oxygen in the water, we saw an incredible increase in the amount of fin beats. So it seemed like they were panting in a way when they had less access to oxygen in the water. So the fins are respiratory. We found they weren't locomotor, but they did have a function in larvae. And then the next question became, well, okay, but is it secondary to just their developing into big fish with nice big fins that are locomotor? Or are they actually adapted to this respiratory function? One of the things we had noticed when we did a lot of our behavioral work was that during abduction, that movement away of the fin, we always saw this bend right at this middle place between the muscle and the rest of the fin membrane, only when the fin was being pulled up, and then the fin straightened and it came down. There's nothing morphological that would suggest that you would get a bend there if you were just looking at the anatomy of the animal. But in fact, when we did some analysis of the curvature along the length of the fin from the base, so from the base of the fin out to the tip, we found that over and over, as we looked at where curvature was high, it was high during abduction in that one area of the fin. Basically, they have this soft tissue joint in the middle of their fin. And this is not something that would occur in the adult, which has a very different morphology. And so we asked whether this was actually could be evidence of a larval adaptation for respiration. So although we thought about it, there was no way we could figure out, or we could go in and either stiffen the fin or somehow change mechanically how it would respond to the fluid forces and its muscle contraction. But we did have our fluid dynamics models where we could change the angle and movement of the fin in all sorts of different ways. So actually we're still doing some of these experiments with some new hypotheses. But the first one was just to say, okay, if we take out that bend and stiffen the fin, do we see changes in respiratory ability? Do we see changes in fluid mixing? And yes, we did. It was not as effective at mixing fluids, so at exchanging oxygen as when you had the bend. So this raised new questions for us, and there are lots of different ones to pursue right now. One is, we think about respiration and locomotion being coupled, or respiration and limbs being coupled in interesting ways. And it had never really been thought about in fish. But a big question that came out of this is how is the system, the pectoral fin, respiration and locomotion linked in larval zebrafish? In particular, is there a time when respiration of the fin is no longer an important thing to do? The fish is too big to respire cutaneously, or the gills develop. Is that when the fins start to be used for locomotion? There's a correlation there, but we don't know exactly how that's working. And lastly, I would say a big question is, did our forelimbs first evolve for respiration as respiratory structures and later become locomotor structures? I think it's an important question we need to ask. Interestingly, when we looked through the literature of zebrafish mutants, we found that a lot of the mutants that have mutant fin phenotypes also have mutant gill phenotypes. So even at the genetic level, there's a clear association between these systems in these animals. So one of the big things we're doing now is using transient transgenics. We're going in to look at individual cells and map out more of the connections. And actually, a big part of that has become looking at mechanosensation of the fins and other types of sensory capabilities of the fins. That work is being done by Katie Henderson and Evie Menelau. I'm not going to talk about it today, but they'll be at the Society for Neuroscience. And if anyone's going to SFN this year, please come by. But in parallel to all the zebrafish work in the lab, a separate line of our research had been working on these robots that I mentioned earlier. And in particular, what the Navy wanted to know was a very sensory question. Like, yeah, we have motors, we can drive the fins, but controlling them is a problem. In particular, sort of open loop control where we're just driving motor outputs, not working very well. What we need to figure out is what information are the fins taking in that makes that appropriate output that can modulate the movements in these animals. So we started working with the Tangora Lab and others on this question, and actually, except for a few papers, mainly from here, looking at the sea robins, no one had looked at the mechanosensation in the fins at all. So tons of us had done movement for our whole careers and never thought about the fins as sensory structures as well. And So a few of my other students, particularly Richard Williams and Nicole Newbarth, started this project. And what they sort of took on a new role in the lab of looking at sensation in my movement lab. And they looked at, again, the pectoral fin. And in particular, they were interested in the mechanosensory structures or nerve structures that were associated with these rays. The rays are sort of the bony elements that they're very flexible but bony, and they extend out into the membrane to support it. Well, as soon as they started staining and looking at the fins more closely, they found, not surprising to us now, but there's incredible innervation in the fins of fish. Each of those fin rays shown up here has massive nerve branches extending out. So this is within a fin ray. They exit the rays and go into the membrane. If we look in the membrane of the fin itself, we see lots of different ending types. There's Bouton-like endings, there are free nerve endings, and other types of sensory cells as well. So clearly pectoral fins, and I would say all fins of fishes, are not only motor structures, but they're also important sensory structures. What are they doing? Again, what are they sensing? To get at this question, we had to develop a new rig, which we were lucky to get advice on from some of the primate somatosensation folks who are in my department. So we had a linear motor that basically connected to a post which could actuate a fin. So here's a fin, and it's in a pool of ringers, physiological saline. We're filming it through this little window, and we're recording nerves through this electrode here. And what you'll see on the next slide is how we're actuating the fin and looking at all that information that's coming from those nerves and is heading back to the spinal cord. So this is what a typical actuation would look like. It's slowed down a bit. But we could, I'm not going to show you all of the ways we could work with the motor, but we could drive sinusoids and normal flapping type of movements, but we could also manipulate it in other ways as well. When we did these experiments, we found that, in fact, the fins have incredible sensory feedback coming back to the central nervous system in fish. So this is driving a sinusoid like you saw in the last video, and all of these are spikes, extracellular recording. We could spike sort and see there are multiple different types of fibers sending signals up in response to fin ray bending. Right, so whenever those fins are moving, lots of information is going back to the central nervous system. What is it reporting on? This hadn't been studied before, other than some work that had shown that prion can sense touch. In primates, we know from our own arms and limbs that we have an incredible sense of our movement of our limbs. We can close our eyes and move our hand in space and know exactly where it is. We can touch our finger to our nose with our eyes closed, with no visual input at all. And that's due to all these mechanosensors in the muscles and joints and skin of our arms. Well, what can fish sense in their fins? It turns out that they actually have the same capabilities that we do. So when we would bend, for example, the fin to different amplitudes, so different amounts of bending, we could see increases in activity. So the fin is actually giving information about the amplitude of movement, the extent of movement of the fin. In addition, if we change the rate of the bend, shown here in these ramps, we would change the activity, so they're sensing the speed of movement. And it's harder to see on these slides, but if we bend to higher bend amplitudes, shown up at the top, we'd get activity sustained through the bend. So that would be information about the position of the limb. So just for us, like proprioception really is reporting on movement and position. Fish also have the information that they could use, if they're set up centrally to do it, to interpret movement of the fins and their position in space. Unlike the larval zebrafish, when we did experiments to determine whether this actually made a difference in locomotion, we found, in fact, that it did. So this is a bluegill sunfish, and intact animal here, and one that we've denervated on this side. I can show these. Why don't I just show you with my arms instead every time? So in the intact fish, the fins extend and they adduct all the way to the body and then extend back again, and it's a fairly slow frequency. After the transection, they would still extend way out, but they would only come partway back to the body, so they would stay splayed along the side of the body like this, and they would move faster, so a higher frequency of movement. This is analogous in all of those ways to humans who have a large fiber neuropathy, where they've lost mechanosensation in the limbs. And what you'll see is a stabilizing through a more sprawled gait that stays extended and a higher frequency, so shorter steps and faster steps. And these data are just the evidence for that. Right. So fins are highly mechanosensitive. They seem to use fins. The information modulation can be used the way we use it from our own limbs. And we wanted to play with this question more, with the mechanosensation and the fins, and really understand it through diversity. One way to test whether, and Alejandro and I were talking about this early, whether a feature of a system is fundamental to how it works, or whether it's just a unique capability that arose once, is to test it in multiple evolutions, independent evolutions across a phylogeny. And so what we were interested in doing is to understand how the mechanosensation relates to the mechanics of the fin. And the way we figured out that we could test that is through looking at evolution of mechanics at different places on the phylogeny of fish, and then relating it to the activity of these nerves. So here are a few people who are very involved in this work. Brett Aleo in particular, Bailey and Adam have both helped on this project, but are mainly focusing on touch right now. The group we chose to work on are the wrasses. You guys have some wrasses here. Tautogs, the blackfish, are in this family of fish, as are cunners. Great animals. They're more colorful when you get to the tropics. They're circumtropical groups, so they're nice to work on because it's usually beautiful warm water where you're studying them. And as you can see, their body shapes are very diverse. And that body shape diversity is related to the diversity in how they use the reef as an environment. One of the big differences that you see across these groups is in the type of swimming they do. So a number of wrasses have developed a really efficient flapping flight. In a sense, they're flying through the water. They're using those same aerodynamic properties of birds and other flying organisms. They're flappers. They have very stiff fins in order to accomplish that type of movement. In contrast, a number of wrasses are rowers. They have very flexible fins, and they're basically pushing water around. They're moving slowly with their fins through the water. Mechanically, so they're very flexible. The rowers, we have stiff-finned flappers, and we thought, well, if we can look at multiple evolutions in the system, we'll get a sense of how the mechanics are really related to mechanosensation. So we did a phylogeny, and this is also with Mark Westneat, who's my husband, who's a wrasse phylogeneticist, so that was very convenient. And identified 4 different groups where we had independent evolution of these stiff-finned flappers. And our question was, when you get a stiff-finned organism, do you see a consistent change in mechanosensation that seems to be correlated to that mechanics? And in fact, we did. So, and again, Brett Aleo did a lot of these experiments. He used that physiological rig I showed you earlier to bend these fins and to look at the neural output from the fins. And what he found was in the stiff-finned animals, even at very low fin bending amplitudes, just bending them a tiny bit, you could already see neural activity, neural reporting on that movement. And it peaked out at lower speeds before the fin could get damaged by overbending. In the flexible finned fish, at low bend amplitudes, he saw no response. It wasn't until you got up to higher amplitudes that we started to see activity, and then it ramped up and the same as the stiff-finned fish. So basically, we took this as, it's actually in the literature, the first evidence, at least in vertebrates, of a neuromechanical tuning. We would argue, we're arguing that the mechanosensory system is evolutionarily tuned to function optimally for the mechanics of the actual fin. These guys, their fins are bending all the time at these low amplitudes. That's not really relevant movement, but they need a lot of information on this higher bending amplitudes. In contrast, the stiff-finned animals need more information at very small bends. Some feedback we got, this came out in PNAS, but some great feedback we got from reviewers was, well, couldn't it all just be plastic? And so we did experiments to feel comfortable that it isn't plastic. We don't think that it is. When we looked at two of these pairs of stiff-finned and flexible-finned animals, so here's one pair of closely related fish, and here's another pair down here, what we found is that they didn't fall along one line, right? There were phylogenetic differences in the minimum bending amplitude that you'd get a response. It's just in both of these groups, you would see a decrease in that minimum bending amplitude in the stiffer finned animals. So there's maybe some plasticity going on, but there's a larger phylogenetic signal happening. So now we're taking those robots that I've worked on with James, and we're putting strain sensors along them, and they're providing terrific information that's helping us do closed-loop control of the robotics and be able to control the fins in very, very precise ways. So it's been fun not only to be able to do the basic science work and to understand a bit more about how these organisms work and how the nervous system works, and even how evolution works a little bit, but also to actually be able to build stuff and implement. I guess I would end saying, that MBL is all about diversity. There's so many interesting questions out there in the ocean to study, and we're coming out of this period where a lot of us just work on model organisms. So I think it's just such an exciting time for the students to be starting here and to be thinking outside the box in terms of the animals that you can work on. So I'm going to end there, and we'll thank I thanked all the people who did direct work on this. The only person I wanted to add, although it isn't part of the work that I presented today, is Marco Horb and the National Xenopus Resource, which is at MBL. He's hosted some of my students while I've been in the director position, and is a complete unsung hero of the MBL, because even though he's associated with Xenopus, he's helping everybody else get high-level genetic molecular techniques going in all sorts of weird animals. So he's just been a terrific asset for everyone, including us. And I'll end there. Thank you very much.
Really great talk. Thank you very much.
Thank you.
You've mostly studied carangiform fish?
I'm sorry?
You've mostly studied carangiform fish, is that correct?
Carangiform fish, yeah.
Have you looked at any of the, for example, knife fish or fish that aren't carangiform swimmers?
Right, so not the carangiform group like look downs, but so carangiform swimmers have a stiffer front of their body, and then a certain part of the back of their body tends to move more. Interestingly, George Lauder, who studies a lot of fish swimming at Harvard, we were just presenting this at the Navy a few weeks ago, and he showed some midline traces of carangiform, anguilliform, tuniform. When you actually look at the traces, there's not as much difference among them as one might think. The head tends to point straight, and you actually are getting a bit more. So we have looked at different species of fish, particularly we've looked at longer larvae that tend to be more anguilliform. But right now, to me, it's looking like those classifications probably aren't as diverse as we originally thought. George does a little quiz where he puts them all up in front, and it's like, which one's the eel? Which one's the tuna? And a room full of ichthyologists couldn't get it right. But yeah.
I'm not sure if I understood these interesting experiments where you use the morpholinos to stop fin movement. I still don't understand if the fins are providing oxygen, what happens to the oxygen supply in the fins that can't move there?
In the morpholinos,
those animals.
Yeah.
And also if the fins are providing oxygen, but they stop moving at rapid swimming when you need more oxygen, what's going on?
Yeah, great questions. So the morpholino fish, it's the morpholino to FGF24. So they don't have fins at all. So they don't have passive fins, they just have no fins. And what we found in behavioral studies of those animals was that they tended to move more and to do bigger movements, like they were readjusting in their tank. So we think that they were using axial behavior to break the boundary layer and move to better water when they didn't have the fins. And now we have actually mutants with no fins, lines that do fine too. So clearly it's not you need them or you die. It's more of an intermediate. They're useful, they're important, but you can get along without them adapting your behavior. For the second part of what you asked with fast swimming is a great question too. So with fast swimming bursts, what we noticed, and really this could be, we haven't published it, it could be documented, should be documented by others, was that when you get these fast bursts, often the fish will swim quickly and then it'll rest. And when they're at rest, they're often using their fins. So it may be that the resting, which has often been attributed to muscle physiology, is partly in order to respire. However, as someone who does fluid dynamics, I would also say that when the fish are moving more rapidly, you're getting bigger tail movements, bigger curvature, you're better able to break up the boundary layer. So there's less of a problem of, that diffusion gradient along the skin in fast swimming than in this slow swimming where the body is relatively straight and the body is moving more slowly. So Reynolds numbers are going to be lower because velocity is part of that Reynolds number. I'd be happy to talk to you about it more.
Hi, over here. Oh, hi. Okay, I just have a quick question. So I see you worked a lot with within the osteichthyes group. So I was just wondering what you would think. Do you think there would be any mechanosensory system in chondrichthyes at all?
Yeah, actually I didn't mention it, but there's one of the earliest works looking at fins was in sharks. And they weren't looking at the same sort of bending, but they were looking at pressure sensation and identified it in sharks. So again, it's an older study. There are only a few traces out there, but they definitely have mechanosensation. Yep. And actually, I would say, we've looked at other fins too, and we've looked at a diversity of fishes. All fins are doing it, I would bet, uniformly across all fishes. I think it's a universal feature of limbs that they can't just move. They need to have sensation as a part of them as well.
Am I missing somebody? Oh, sorry.
Oh.
So initially you showed the fish switching from fast movement to slow movement and how the fins got started as they slowed down.
Yeah.
Have you guys looked at central pattern generators similar to the salamander that work in that transition?
We're trying to do now. So there's actually, as part of my postdoc works, maybe Steve mentioned, we do know from some of that work, mainly with Dave McLean, who's now at Northwestern, that there are different populations of spinal interneurons that are used for fast and slow swimming. And it's actually this dorsal-ventral gradient along the body. So you can see a wave of activity of these excitatory interneurons. For the fin movement, so the fast swimming is excited from what we've done and from other work by this descending activity from, in our case, what looks like rostral hindbrain. And so you need that excitatory drive to get fast swimming. when that stops and they move into slow swimming, the central pattern generators in the spinal cord are taking over, and that's all it takes to get that rhythmic movement in the coordinated with fin movement in slow swimming. So there's definitely both in the spinal cord sort of a gradient of CPG sort of circuits, and then there's the switch between brain control driving the activity to it being a spinal CPG set.
Thank you.
Thanks.
Do we understand from this kind of study that just as the human body maps a sort of sensory homunculus, that it looks like the fish may map a sensory, pescunculus?
I love that, pescunculus. Absolutely. Well, so they don't have a cerebral cortex. So if we're not talking at that level, but we're talking about mapping in the spinal cord, they absolutely do. So there's a map that relates to the speed of movement that I was just mentioning, but we also see the maps to morphology. So interestingly, in development, these fins in the zebrafish that you saw flapping, as the animal, and this is true for all fish, as the animal develops, the orientation of the fin changes. So what we think of as dorsal and ventral now are actually flipped. It's almost turning over as we develop the bones and muscles. And so in the spinal cord, what you see is a reverse orientation of the sensory pools, so that the more ventral pools in the spinal cord are connected to the, of neurons, are connected to the fibers that are on the more dorsal part of the fin, and it has to do with this transition. But there's definitely coordination and mapping across sensory, motor, and I would say in terms of speed of movement for the interneurons as well. Yeah. In the brain, we don't know. It's a fascinating question. There are maps of excitatory and inhibitory columns of cells that seem to be related to different types of behaviors. But in terms of the pescunculus, I haven't seen that yet. Great question. Oh.
More students?
There's one behind you.
This may be really ignorant, but do the fins get stiff maybe because they either need or not needed for respiration when the fish is in a developmental stage, and so then it behaviorally develops a kind of swimming? Or did it have a kind of swimming that led to the development?
The mechanics of the fins, yeah. I would argue the latter. So the fin rays, that bony structures in the fins, don't develop until after the gills come online and the membranes and fins are no longer needed for respiration. So it isn't until these later stages of development that we start to see the structures that lead to the differences in mechanics. Oh, genetically. Yeah. Genetically, I don't know. That's a good question. Yep. Yeah, although I would say that we see, yeah, that's a great question. We should look at that for sure.
All right, I like ending questions on that note.
Great. So thank you, everyone.
Thank Melina again. And please join us in the Miggs room for a reception.